Okay, you may start now.
Good afternoon po, Doc Nemi. Nathan and I today will present the genetic basis of booster doses for newer COVID variants in the Philippines. So for the outline of this presentation, we will focus on the SARS-CoV-2, especially the genome and proteome of SARS-CoV-2, the COVID-19 variants such as the Alpha, Beta, Delta, Gamma, and Omicron, the vaccination, which are the vaccines available in the Philippines and their mechanisms, the booster dose mechanism, and lastly, are booster doses for newer COVID variants okay? So we'll focus on the antigenic properties, the antibody escape, neutralization escape, the current evidence on booster efficacy against Omicron, and suggested booster dose or improving currently available vaccines. So start for the genome and proteome of SARS-CoV-2. This variable origin of transcription allows the SARS-CoV-2 virus to encode for around 50 proteins that have non-structural, structural, and accessory functions. The SARS-CoV-2 is a single strand of RNA with a positive strand. It has a 29,903 base pairs. And again, it has at least 50 different sites where translocation can begin. On the initial two-thirds of the RNA sequence, it can encode the two main transcriptional units, the ORF1A and ORF1AB, which encode two polyproteins, the PP1A and PP1AB respectively. The larger PP1AB contains ORFs for at least 16 non-structural proteins, and it is important to note that the non-structural proteins have various functions in biological phenomena that are important for the virus, such as replication, the correction of replication errors, or what we call proofreading, the translation, the suppression of host proteins, and the immune response blockage and RNA stabilization. And then for the final third of the RNA sequence of the SARS-CoV-2 virus, it encodes proteins that define the structure of SARS-CoV-2 as well as accessory proteins. And then the accessory proteins are not significant for viral replication, but however, they play an important role in interactions between the virus and host, including modulating and blocking the production of pro-inflammatory cytokines. The 8 accessory proteins and 4 major structural proteins, including the spike protein, the envelope protein or E, the matrix or membrane protein M, and nucleocapsid protein N, are located in the 3 terminus of the SARS-CoV-2 genome. The SM and E proteins are embedded in the outer membrane and give the virus its distinct shape and structure, while the spike protein plays a very essential role in binding to receptors on the host cell and is crucial for determining host tropism and transmission capacity, mediating receptor binding and membrane fusion. Inside the virus particle, the RNA is tightly coiled and coated with the fourth structural protein, the nucleocapsid, which the purpose of it is to protect the genetic material of the SARS-CoV-2 virus. And proceeding, we will now tackle the COVID-19 variants, such as the previous variants of concern, the Alpha, Beta, Gamma, Delta, and Omicron. So, the previous variants of concerns, there are 4 main receptor binding domain or RBD amino acid substitutions, which have been the focus of studies that assess virulence and immune evasion. First is the N501Y, which is found in the ACE2 binding site of the receptor binding domain. And the second and third substitutions are E484K, Q or A, and K417T or N. And lastly, the L452R. So these 4 have been the focus of the studies of the previous variants of concern. So first of all, what is the alpha variant or what we call the B.1.1.7? It was first detected in the late September 2020 in the United Kingdom and quickly became the predominant strain in the United Kingdom. The first case of the first case of alpha variant in the Philippines was detected on January 13, 2021. So medyo matagal siya na-detect in the Philippines, then it was detected firstly in UK. So the alpha variant was associated with an increase in severity and consequently a rise in mortality rate of up to 61%. And studies have shown that the N501, which is also found in the beta and gamma variants, contributed highly to the increased transmissibility and higher viral burden of the alpha variant. So the asparagine at the position 501 or N501 is one of the 6 essential amino acids involved in the ACE2 receptor binding, and its substitution with tyrosine or Y has been shown to increase binding affinity to the host receptor. It then contributed highly to the increased transmissibility and higher viral burden of this alpha variant. Also, it contains the P681H substitution, which is found in an area adjacent to the furin cleavage site that is known to be important in infection and transmission. And lastly, the alpha variant has the DH69D70 deletion, which is also present in other variants, and this enhances viral infectivity in vitro and has been linked to certain commercial kits failing to detect the S-glycoprotein. So basically yung DH69B70, ito yung nag-increase ng viral infectivity. Tapos parang nagkaka-problem ka with the detection of the S protein because of this mutation. Next. The beta variant or the B.1.351 was first detected in October 2020. It became the dominant strain, resulting in the second wave in South Africa. The first case of beta variant in the Philippines was detected a month after the detection of the alpha variant. This variant has increased transmissibility and virulence, parang alpha variant compared to the wild type of the SARS-CoV-2. And then the amino acid substitutions in the beta variant S protein are more extensive than in alpha, presenting 10 changes with 3 in the receptor binding protein. So yung 3 na changes sa RBD mo is yung N501, the K417N, and then the E484K. So the purpose of the N501 residue mutation is important again for the ACE2 interaction. Because of this mutation, mas madali kang bind with the ACE2 receptor mo. It is also same with the alpha variant. Next is the K417N and E484K. These are not predicted to play a major role. However, these two residues may in fact reduce the binding affinity, which shows that the increase in transmissibility seen in beta variant is primarily due to the N501, which is also present in the other variants. So next, the gamma or P.1 variant was detected in 4 Brazilians traveling to Japan in January 2021 and was responsible for the resurgence in infections in Manaus. So in recent times, parang less than 20 cases lang yung nag-result sa atin sa gamma variant. It was first detected on March 2021. It is also parang almost same time siya with the beta variant, but the beta variant was more predominant than this gamma variant in the Philippines only. So this contains 17 amino acid substitution, including a similar trio seen in the beta variant. It is the N501Y, the K417P, and E484K in the S protein found in the beta variant. And similar with the beta variant, yung N501Y lang yung nagpe-play ng major role in the increase in transmissibility and infectivity of the virus. So next, the Delta variant or the B.1.617.2. It was first detected in December 2020. It was responsible for the massive rise in cases causing a second wave in India and breakthrough infections in multiple gatherings in America, as well as in the Philippines. The first case of Delta variant in the Philippines was on May 2021, but the first confirmed local case transmission was announced on July 16, 2021. So 2 months after the first detection. So this dominant variant has 8 S protein changes. It has an amino acid substitution found in the RBD, the L452R, and one in the S protein furin cleavage site, the P681R. And these have been implicated in the increased transmission associated with this variant. The P681R substitution, also found in the alpha variant, was demonstrated to increase fusion activity to the host receptor, which could possibly increase infectivity and transmission rates. And also, the Delta variant is more transmissible than the original strain, as well as the previous variants such as the Alpha, Beta, and Gamma. And then studies have shown that the risk of hospitalization was also found to be double that of the Alpha variant, with the recent study indicating that hospitalized patients with infected of Delta variant had more severe disease and a higher in-hospital mortality rate. So ang sinasabi ng mga previous studies basically is the Delta variant is more double the transmissibility and virulence of the previous variants as well as the original strains or the Wuhan wild type. Pero after ni Delta variant, 1 month after lang, nandito na si Omicron variant or the B.1.1.529. So it was first reported on November 24, 2021 in South Africa. So compared to the other variants, this Omicron variant has as many as 37 mutations in the spike protein, 15 of which can be found in the receptor binding domain, and as many as 25 of such mutations being almost unique to this new variant. So sobrang laki ng difference ng Omicron variant compared to the previous variants. And later we will tackle, sino ba yung kamukha ni Omicron variant? Kanino ba siya basically nag-stem from? So the Omicron variant has increased transmission compared to the original SARS-CoV-2 virus, as well as all the previous variants such as the Alpha, Beta, Gamma, and Delta. So again, sabi ko nga yung N501Y can be found in all of the strains, all of the variants. So it increases the binding to the ACE2 receptor, which increases transmission. And then the combination of this N501Y mutation and the Q498R increases the binding affinity even more. So mas malala talaga tong Omicron variant. Mas marami siyang mutations, tapos double that of the other variants pa yung transmission capability niya. So the H655Y is proximal to the furin cleavage site and may increase spike cleavage. So I didn't put it here in the presentation because the H655Y was found to be irrelevant pala. So sinasabi na naman ng studies na si N501Y talaga yung may pakana ng mga increase in transmission, as well as sa Omicron, the combination of the N501Y with the Q498R. And then meron pang isa na from the earlier study sinasabi na it can contribute, but however the recent study said na si N679K wala rin pala siyang purpose. So may mutation lang siya pero hindi siya nagko-contribute sa increased transmissibility of the variant. Pero lastly, si P681H, it can enhance the spike cleavage, and this spike cleavage, the enhancement of this, could aid transmission. So this mutation is also found in the alpha variant, and an alternate mutation at this position is found in the delta, which is the P681R. So etong P681H, actually ito yung mutation na nagbibigay ng hint kung sino ba yung kamukha talaga ni Omicron variant compared to the other variants. So here's a summary table of the emerged COVID-19 variants. So the alpha variant, ito naka-highlight yung mga RBD mutations. So alpha variant has the N501Y only. So the sequence neutralizing potential, which is calculated in folds, So decreased by 2 dito sa alpha variant, and then the infectivity is increased by 1.5. While sa beta variant, yung neutralizing potential niya is decreased by 8. And then yung infectivity niya is increased by 1.5 as well. While dito sa gamma variant, yung neutralizing potential niya was decreased by 5, and then yung infectivity niya was increased by 2. Tapos sa Delta and sa Omicron, may kita mo yung huge difference ng decrease of neutralizing potential, which can infer na mas grabe talaga mas mabilis talaga siya mag-spread kasi mas nakakapag-evade siya from the immune responses. Since sa Delta, yung neutralizing potential niya in terms of folds was decreased by 6 or 8, and then infectivity niya can increase by a maximum of 2.5. Sa Omicron, very, very huge yung difference niya since the neutralizing potential was decreased by around 10 to 40. So, grabe yung difference niya compared to the other previous variants of concerns. And then yung infectivity niya was increased by 2.5. So, next, I put lang a summary table lang of the effects of some frequent mutations found in the spike protein gene of the SARS-CoV-2. So, if makikita natin, yung N501 here, it really can increase transmission plus decrease neutralization. So, basically, this table states na yung N501 as compared to as supported by the previous evidence, the recent studies, N501 talaga yung may pakana ng mga increase in transmissibility ng previous variants. Pero you can also check here na marami pa ring mga mutations that can contribute dun sa decreased neutralization and increased transmission of the variants. So next here, dito we can compare the differences in nucleotide decomposition between the variants, and as well as the percent identity, the number of gaps, and the number of identical nucleotides in the genome. So we'll start first with the first table. So makikita natin dito, the alpha variant had the greatest identity rate with the Omicron variant at 99.63%. And furthermore, ito yung Omicron variant, yung pinakauna, it has or showed the greatest number of gaps during genome alignment with other viruses, ranging from 43 to 63 gaps. So based on the sequence alignment differences and gaps, it is proven that the alpha variant has the fewest nucleotide alterations when compared to the Omicron variant. Hence, sila yung may closest relationship. So basically, according to this table, Omicron variant most likely stem from the alpha variant. So this close relationship between Omicron and the alpha variant may indicate that the Omicron variant was in circulation already for a lengthy period before it was already discovered. So baka nahirapan lang siyang ma-detect agad since it is very, very alike with the alpha variant. Remember, alpha variant started no late 2020. Omicron was detected no early 2021. So baka within those months na dominant yung alpha variant, nandun na pala si Omicron, hindi lang siya agad na-detect since very alike siya with the alpha variant. And this, I showed this beautiful picture of the phylogenetic tree of the SARS-CoV-2 virus. So regardless of the specific mechanisms promoting the emergence and selection of mutations in the viral genome, what is now abundantly clear is that SARS-CoV-2 has mutated a huge number of times. Since it was originally discovered in 2019, around 3,590 mutations already. Yan na yung mga total number of descendants from the original Wuhan type 2019 SARS-CoV-2 until the recent COVID-19 variants. And this will continue to do so in the near future according to the study. And this picture states that the virus again has already originated 3,590 times including the Omicron recent subvariants, and its subvariants will most likely become the more prevalent worldwide in the following months due to higher infectivity. So we can see naman ngayon, the recent variants such as the BA.4.5 and the BA.2.12.1, sila yung mga predominant subvariants now. And we will see later kung paano ba sila nakaka-invade sa mga immune response. Bakit sila variants of concern now? Kasi as of the moment, silang tatlo lang yung variants of concern. Other variants are now called VOMs or variants under monitoring. So next, the Omicron subvariants or the BA.2, 0.12.1, the BA.4, and then the BA.5. So this one's a good Venn diagram comparing the BA.1.2, 0.1.1, and BA.3. So ito yung mga older subvariants before we tackle the newer subvariants. So among the 4-week comparison between Omicron and subvariants, the BA.1.1 has one unique mutation, which is the R346K. And then the BA.2 has 8 unique mutations, the T191, L24 deletion, P25 deletion, P26 deletion, A27S, V213G, P3676A, and R408S. And then the BA.3 has one unique mutation, the R216 deletion. And then, yung sinasabi dito na yung BA.1, wala siyang unique sa kanya na mutation, only commonly shared. So, ito yung pinakaunang subvariant. So, yung BA.1, parang almost alike siya with the original Omicron variant. So, in comparison to the receptor binding proteins, or the 319541, there are 11 commonly shared mutations. So medyo marami, so hindi ko na i-mention isa-isa. Pero if we're looking back dun sa Venn diagram, yung 11 as yung mga naka-highlight in bold under dun 21. So if you count that, 11 siya. So yun yung common niya for all. And then next, BA.1.1, ayun nga, has one unique. BA.2 has two unique mutations in terms of the RBD, or the receptor binding domain. And then the commonly shared mutations between BA.1 and BA.1.1 are S371L and G496S. And then Y505H is the commonly shared mutation among BA.1, BA.1.1, and BA.2. And G446S is the only common mutation among BA.1, BA.1.1, and BA.3. BA.2 and BA.3 has two commonly shared mutations, the S371F and then the D405N. So, going back, the N501Y mutation, which enhances receptor binding strength, which is present to all previous variants of concern such as alpha, beta, gamma, and delta, and Omicron. Present din siya ngayon sa all 4 subvariants ng Omicron, which is the BA.1, BA.1.1, BA.2, and BA.3. And this N501Y mutation has a general positive relationship dun sa stabilizing effect niya on the receptor binding and then their population incidence. So again, this study has proven na N501Y talaga yung may pakana nung increase in transmissibility and infectivity ng subvariants of the Omicron virus. So big problem talaga yung N501Y mutation. So next, dito, ang tanong natin, saan ba nanggaling si Omicron, yung bagong newer variants, the Omicron BA.2.12.1, BA.4, and BA.5. So, this study has shown na ang kamukha niya pala si Omicron BA.2. And this Omicron BA.2 contains 3 additional mutations, including the 376A, D450N, and R48S, and it lacks the G446S and G496S, which is present in the subvariant BA.1. And also, the S371L on BA.1 is substituted with S371F in BA.2. And this Omicron BA.2, if you can remember, it is also called as the stealth variant. So bakit siya stealth variant? Because it does not have the amino acid 6970 deletion. So which is yung nakita natin sa mga previous variants of concern. So this Omicron BA.2, wala siya nun. And yung purpose nun, di ba, nakalagay ito sa table kanina, was S-gene dropout. And ano meaning ng just S-gene dropout or S-gene target failure? It is meaning not recognized by the SGTF, which leads to the complication in tracking its transmission and why it is called stealth variant. So mahirap siya matrack. Siya yung variant na pinakamahirap ma-recognize because missing nga siya no 6970 deletion. So yun yung naging prominent sa kanya. And these three na newer variants, kamukhang-kamukha nila si Omicron BA.2. It has identical RBD sequences to BA.2. However, meron lang mga additional mutations which can be unique from the BA.2, pero in reality, magkamukha lang sila halos. So, yung addition nila is sa L452 and F486 substitutions, namely sa BA2.12.1, the unique one sa kanya that yung una mo dapat hinahanap kung gusto mo siya ma-differentiate from the Omicron BA.2 and other newer subvariants is the L452Q. While sa BA2.13, it's L452M, and then sa BA.4 and BA.5 is L452R, as well as F486V. So this is how you can differentiate them from BA.2 and differentiate among themselves. So ito yung mga hinahanap nila when na mutation para ma-distinguish yan. So coupled with the spike structural comparisons, BA.2.12.1 and BA.4 as well as BA.5 exhibit comparable ACE2 binding affinities to BA.2. Dahil nga magkamukha sila, almost same lang din yung mechanism nila. However, these newer subvariants displayed higher transmission advantage over BA.2 because of the additional substitutions present. So, ang sinasabi, magkamukha lang talaga sila, pero dahil dun sa additional substitution, mas naging higher lang yung transmissibility niya, but not really the virulence.
Thank you for that, JM, for discussing a very comprehensive genetic basis for each of the COVID variants. Now we will briefly tackle COVID-19 vaccination, so the vaccines available in the Philippines, and mechanisms of booster dose vaccination. So next slide, please. Here's a very nice publication material from DOH. So these are the vaccines available in the Philippines, updated as of June 2, 2021. As of this point in time, on June 2, 2021, only 7 vaccines had received EUA or emergency use authorization from the FDA. Since then, there have been 2 additional EUAs issued for Novavax on the farthest right column here, and the inactivated varicella vaccine produced by Sinopharm. So now let's discuss some of the mechanisms of each or the basis for each vaccine. Wait. Skipped a little bit, but that's okay. Okay, here's the inactivated. So these inactivated SARS-CoV-2 viruses include Covaxin produced by Bharat Biotech, the varicella-based vaccine produced by Sinopharm, and Coronavac produced by Sinovac. Briefly, these vaccines are based on inactivated or killed version of the SARS-CoV-2 virus, which are used to trigger protective immunity against the COVID-19 virus. On the other hand, the viral vector-based vaccines include your Sputnik Light and Sputnik V, produced by the Gamaleya Research Institute. The AD26-CoV-2S vaccine produced by, produced by Janssen Pharmaceutica, a subsidiary of Johnson & Johnson, and the ChAdOx1S and COVID-19 recombinant vaccine produced by SK Bioscience and the and by the Serum Institute of India, the latter of which is marketed as Covishield, and both these vaccines are developed by your AstraZeneca. Next slide. And for the protein-based vaccines, the singular one approved for use in the Philippines by the FDA is Covovax, produced by Novavax. It is a recombinant spike protein nanoparticle vaccine, which essentially uses pieces of the COVID-19 virus, such as fragments of the S protein, to induce an immune response. And lastly, the mRNA-based vaccines available in the Philippines are your well-known vaccines produced by Moderna and Pfizer, designated as mRNA-1273 and Comirnaty or BNT162b2, respectively. And briefly, these vaccines use mRNA packed in lipid nanoparticles to induce cells to produce antigen proteins coded by the mRNA and to stimulate an immune response. Here is an excellent graphical abstract depicting the various vaccine strategies for COVID-19. So here we see the protein subunit vaccine, the mRNA vaccine, the viral vector vaccine, and lastly, the inactivated vaccine. Now for booster doses.
So for the booster dose mechanism, it's a brief discussion now. So what is a booster and how does it improve immunity? So a booster is a dose which refers to a single dose of COVID-19 vaccine given after the protection provided by the primary series has begun to decrease over time. So basically, a booster dose helps maintain the immunity for longer periods. So kapag yung primary series mo, which is the first two doses, after quite some time, I think 4 months yung ngayon, that's when you can get a booster. And then, so basically the WHO recommends kasi that it is best, people are best protected when they stay up to date with their COVID-19 vaccines, which includes getting all recommended boosters when eligible. So how does it improve immunity? So they stimulate lang the individual's immune system to produce additional antibodies and memory B cells and T cells. So the purpose of this booster is, so from the primary series, may exposure ka na. So you just want to add additional exposure. So para mas mag-produce ng additional antibodies yung immune system mo. However, in some individuals kasi, hindi sila nagkakaroon ng especially the immunocompromised ng antibodies presented from the primary series. So this booster, parang ginagawa niya for those individuals nga na hindi nagpo-produce ng antibodies from the initial primary series. So additional siya to stimulate the immune system. So in the Philippines, the first booster now is open to the elderly immunocompromised as well as the general public. But we will see later if recommended ba ang second booster shot to the general public now.
Thank you for that segue. So now I'll discuss booster doses for newer COVID subvariants as mentioned by JM earlier. These are your BA2.12.1, BA4, and BA5. So next slide. Just want to set the stage again that the global spread of the SARS-CoV-2 variant Omicron has strained healthcare systems worldwide and posed a challenge to the efficacy of COVID-19 vaccines. During the recent surge in January 2022, that the DOH reported that Omicron made up the majority of cases, specifically the sublineages BA1 and BA2 compared to the Delta variant. So significantly outnumbered the Omicron and Delta variant during the DOH sampling, which is indicative of Omicron's higher transmissibility as extensively discussed by JM earlier. So next slide. However, in the last 2 months, the DOH has reported the detection of Omicron sublineages BA2.12.1, BA4, and BA5 in the Philippines. These sublineages are suggested to have higher transmissibility compared to BA2, the Omicron sublineage that dominated the previous surge, which I will begin to discuss why in the next few slides. So in this study already cited by JM earlier, they found that BA2.12.1, BA4, and BA5 display stronger neutralization evasion compared to BA2, as depicted by the larger reduction in plasma NT50, or rather the number of plasma dilutions required to destroy 50% of the virus. What this basically means that the more it decreases, the less antibody response that the plasma had. But it is important to note that in this study, the aforementioned sublineages were also compared against SARS-CoV-2 with D614G. It's mentioned in JM slides earlier that it's found in beta, gamma, and delta. It confers increased transmissibility and was first present in Omicron as of now. So it's basically compared to almost wild type, but with an additional mutation. Also, the study used plasma obtained from individuals with 3-dose regimen of Coronavac. Next slide. I would like to highlight. So it used 3-dose regimen of Coronavac, known locally as Sinovac, or 2 doses of Sinovac and a booster dose of ZF2001, which is unavailable in the Philippines, and 3 doses of Sinovac and figure C, and were infected with Omicron BA1. And in figure D, individuals with SARS-CoV-1 infection, the virus that caused the initial SARS outbreak, and 2 doses of Sinovac with SZF2001 booster. So next slide, please. Essentially, the important finding from this study is that patients with 3 doses of Sinovac and infection with Omicron sublineage BA1 may not receive as much protection against the newer Omicron sublineages BA2.12.1, BA4, and BA5. So this study was performed in Beijing and used the Sinovac vaccine. Are there results confirmed by studies in different settings and those that use different vaccines? So next slide, please. Now we move on to this study by Willett, which was performed in the UK and used the ChAdOx1 vaccine produced by Oxford AstraZeneca and BNT162b2 produced by Pfizer BioNTech and the mRNA 1273 produced by Moderna. So what this slide shows is that in figure A, plasma serum from triple vaccinated individuals with 2 doses of AstraZeneca and 1 dose of either Pfizer or Moderna have similar drops in neutralizing titers against all Omicron sublineages compared to wild type. Weakness, there is still neutralization escape or immunity escape. So next slide. But it is important to note that in figures B and C, the 3 doses regimens of Pfizer or 2-dose AstraZeneca with Pfizer booster increased neutralizing titers by more than 10-fold against the newest Omicron sublineages BA4 and BA5, and similarly for BA2 and BA1 and BA2, the predominant sublineages during our January surge. So what figures B and C say is that booster doses still confer some form of immunity against even the newer subvariants. Next slide. What I think the most important and the most interesting finding from this study is that neutralizing antibody titers against BA4, BA5 from triple vaccinated individuals with a breakthrough Omicron BA1 or BA2 infection, so they were still infected despite vaccination. This slide shows that there is still the breakthrough Omicron infection generate a robust pan-Omicron antibody response as demonstrated by the by the maintenance of by the not so large decrease, I mean, in antibody titers for those who received a breakthrough infection after triple dose vaccination. So next slide, please. So infections with BA1 or BA2 provide some measure of protection, and thus this study suggests that booster doses derived from Omicron-derived antigens from BA1 or BA2 may be an effective approach in inducing cross-protective immunity against all identified sublineages of Omicron. Now I'll discuss one more study. Lastly, in this correspondence published in the New England Journal of Medicine last June 22, 2022, neutralization escape of Omicron sublineages BA2.12.1, BA4, BA5 was also found. This study was conducted in patients with 3-dose regimens of the Pfizer-BioNTech vaccine and in vaccinated individuals with breakthrough infections of BA1 or BA2. And again, the important finding, similar to previous studies, is that booster doses significantly increase, neutralizing titers compared to pre-booster levels. But Omicron sublineages, especially BA2.12.1 and BA4/5, display significant neutralization escape. Next slide. I've discussed 3 studies so far, and it is difficult to compare levels of neutralizing antibody titers and plasma NT50 findings across studies due to differences in study population and in method, but one trend remains clear. Whether your vaccine in booster is Sinovac, AstraZeneca, Pfizer, or Moderna, booster doses improve levels of neutralizing antibodies against newer COVID subvariants, but not as well, unfortunately, as compared to older COVID variants. But still, booster doses provide some measure of protection. And now I'll discuss what do real-world studies of a vaccine efficacy against Omicron say. Next slide, please. So this study by Chenchula is a systematic review of the effectiveness of a 3rd dose or booster dose of the COVID-19 vaccine against the Omicron variant of concern. So a total of 27 published studies were identified, 5 in vitro studies, 20 prospective observational studies, and 2 case reports which utilize the AstraZeneca, Pfizer, Moderna, and Sinopharm vaccines. What is important to note is that all of these studies report protection against hospitalization with Omicron or increase in neutralizing antibody titers and association of lower likelihood of symptomatic SARS-CoV-2 infection. In summary, all the findings reviewed from currently available studies support the evidence for booster dose vaccine efficacy against SARS-CoV-2 variants, specifically Omicron. So next slide, please. So to conclude our presentation by me and JM, what is our path moving forward despite the rapid evolution and accumulation of large amounts of mutations of Omicron? I'd like to highlight the finding from the second study I mentioned done in the UK that booster doses based on Omicron BA1 or BA2 may provide immunity against other sublineages of Omicron and possibly future yet unidentified variants of concern. Next slide, please. This is in line with the WHO interim statement on the composition of current COVID-19 vaccines. The WHO also suggests the inclusion of Omicron in an updated vaccine composition, which can be beneficial if given as a booster dose to those who have completed a 2-dose COVID-19 vaccination primary series. And thus, moving forward, booster doses for newer COVID variants should be based on Omicron. Next slide. Yeah, thank you. And I just wanted to mention that Pfizer actually announced just last Saturday an Omicron-adapted COVID-19 vaccine candidate, and they found that in preliminary studies, it demonstrates high immune response against Omicron. So this is where the future lies for COVID-19 vaccine development. Thank you. This concludes our report.
Thank you, Nathan and JM, for a very extensive report, and I'm really very much impressed with the amount of data that you were able to gather, and also much impressed with the way you synthesized the data. And this one is a very important finding, particularly if we are to encourage our countrymen to have their booster shots, no? And so after hearing the talks that the two of you gave, it is very clear that mutations, even though there are a lot, there seems to be common findings or common mutations that really drive the escape from, of course, the neutralization by vaccines, and also the easy transmissibility of the newer variants. So if you were going to suggest in terms of testing, therefore, what would your suggestion be as far as the way to move forward when it comes to testing for the newer variants, and where should we focus our attention when it comes to testing for these variants? Particularly when you talk about the you mentioned about the different mutations, no, that are being studied. Should we focus our attention, especially here in the Philippines, given that we have limited resources, unlike perhaps other countries where they can test all the possible um, abnormalities or gene variants or deletions? Where should we focus our attention here in the Philippines?
So I think based on the mutations provided, uh, nagkakaroon sila ng distinction based on dun sa mutations on the RBD or the receptor binding domain. So I think if one way to distinguish yung mga variants of concern now, I think we can focus on that, yung sequence under the or mutation ng genome under the receptor binding domain po.
Okay. Thank you, JM. One other important thing which I think is very practical in terms of a clinical point of view is regards your discussion on the booster doses of your vaccines. And you showed data, Nathan, about the differences with the effectiveness of the booster vis-a-vis their primary vaccination series. So, if you go back and therefore assuming, say, of course, this has already happened, and in our country we had several types of primary vaccination series, and we know that we started with the CoronaVac of Sinovac, and then eventually we had Pfizer, we had AstraZeneca, and we had Moderna. Given the data that you were able to get, and particularly the effectiveness of the booster doses, which, in your opinion, would have been the best? Okay, siyempre retrospective na lang ito, no? Kasi we cannot bring back time anymore. If we had the benefit of the information that we have now, way back last year when we were just starting vaccination, what would you think would have been the best combination of vaccination together with the booster?
Based on based on the what I've read, and also the data on the booster dose immune response, I think that if we transport us back to last year when the vaccines were just available, I would advise a patient deciding between Sinovac, AstraZeneca, Pfizer, Moderna to go for the mRNA-based vaccines, or if wala AstraZeneca, or if wala din yon, Sinovac. So that's the hierarchy that I would suggest. And for booster doses, I would strongly suggest an mRNA-based vaccine for the booster dose.
And I think that's what we have right now, right? For our booster doses, we have both Pfizer and Moderna, which are both mRNA-based vaccines. And although the current recommendation of the Department of Health is just to limit the booster vaccination to the elderly and the immunocompromised, um, of course, the frontline health workers are also being given booster shots, and I think a lot of even medical students, no, and even a lot of those people who have comorbidities, even if they're not yet senior citizens, are also being or are also want to get the second booster, and so in your opinion, based on the data that you have presented to us, what would be your advice to people, number one, who haven't had their booster shots, and number two, those who already had their first booster, should they actually have a second booster shot?
Doc, parang based on the articles, it says na the primary series as well as the first booster shot, good siya for the newer variants now. However, if ako yung clinician, I will suggest na dun sa mga patients na wala pang booster shot to get their first booster shot, but to wait for the updated vaccine for their second booster shot. Since nga po, diba yung current guidelines for the second booster shot is only recommended for those na hindi nag-elicit ng antibody response, especially for the immunocompromised, and then the elderly, and the frontliners, doon lang yung may evidence na okay yung second booster shot since nag-start dun sa kahit sa first booster shot nila mababa yung immune body response nila. But for the general public, I think it's better to wait for the updated vaccines, which I think they're currently doing it now, yung updated vaccines na Omicron derived. I think it's better to wait for that for the second booster shot.
I agree. I agree with what you mentioned, JM. Would you like to add anything else to what JM mentioned, Nathan?
No, I wholeheartedly agree po na if you don't have your first booster shot, get it now. If you don't have if you're waiting for a second, wait for the Omicron booster.
Okay.
Booster. So Pfizer already announced theirs to the delight of their stakeholders.
I think that's the
But
beauty, of studies, particularly if you are familiar with genetics, no? And what we are doing right now is we are really optimizing and really using our knowledge on genetics to help the healthcare system all over the world. And I guess this is where the clinical application and practical application of genetics comes into real, very clear utility for the health of everyone around the world, not just here in the Philippines. So any last words before we end our session this afternoon? Perhaps we can start with JM, then we'll have Nathan.
Actually, Doc, I mean, in the recent pandemic days, I realized na how important nga yung knowledge or kahit foundation sa genetics since it's easier to understand yung mga current events now kung bakit patuloy pa rin na patuloy nagpo-produce ng variants, how different it is from the previous ones. And then next is I would like to thank lang po for the opportunity for this presentation kasi I mean, hindi naman po namin malalaman extensively yung mga nangyayari sa current events if we didn't do this presentation. So thank you po, Doc Naimi.
Thank you, JM. Nathan.
Before I give my thoughts, I'll just share that I was wondering why sobrang lagi may focus on the elderly, the immunocompromised, and syempre, okay, yes, their immune systems are weak. But I read thank you for this opportunity for us to read up. I read that Omicron was found to have very large jumps. There's a lot of mutations, and it doesn't occur now in immunocompetent individuals. It is theorized that mutations that cause very large trend jumps in large numbers of mutations in infectivity, transmissibility occur in immunocompromised patients who have had the chronic infection for a long time. So the virus has had time to replicate and mutate. So dun po nanggagaling. And thus, I guess, what I mean to say from all of this is that I think genetics has a very strong clinical, if understated, importance in terms of how we see things, see diseases, see people, see health policy making. Yan. And thank you for this opportunity ulit, Dr. Nemi.
Thank you to both of you. Actually, I really am pleased that you were able to appreciate this particular report that you made. You learned a lot from it. Very practical, very helpful. If people will approach you anytime, you can give them the advice, no, based on science, based on what you have just reported to us. You can be good advisors to people who will ask you should they get a booster, and who will be the best candidates to get it. So thank you very much to the two of you. Since this session is recorded, I can share your report to other people, and of course to other faculty members, and I'm just so happy that you gave an excellent report, the two of you. And given such a short notice, no, less than a week lang ang inyong preparation, but yet you gave an excellent report. So congratulations, JM and Nathan. And so we'll be ending our session this afternoon. And once I have the recording, I can give you a copy, both of you, para you can go back to your report and share it with your classmates as well.
Thank you po,
Thank
Dr.
you,
Nemi.
Dong.
Thank you. Excellent job.
Doc,
Congratulations.
may we request a picture lang po before we end?
Sure,
Yes.
sure, of course. Oo, mag-picture
I'll
tayong
take the
tatlo.
picture na lang po.
Sige, okay.
1, 2, 3. Ay, again po pala. So, 1, 2, 3. Ayan po.
Okay, sige. At least makakapagpahinga na kayo tapos na ang inyong academic work.
So, selling for JM.
Meron pa ba?
Yeah.
Meron pa ba kayong report? Wala